Okay, so these are the instructions for the gastric digestion lab. This is day 1 of 3 of the digestion labs. So you'll be following along with the gastric digestion handout. I'm going to read the intro, the background, and the procedure, and then I'll show you guys how to do the procedure. So, in this exercise, you will study one of the processes of digestion. Follow the instructions below, observe the outcome, and answer the questions that follow. Food travels from the mouth to the stomach for gastric digestion. During gastric digestion, glands in the stomach wall secrete gastric juice, which mixes with food particles. Gastric juice is a mixture of hydrochloric acid and the enzyme pepsin. At body temperature, meaning 37 degrees Celsius, and in the presence of hydrochloric acid, pepsin reduces proteins into amino acids. The food particles are converted to a semi-liquid state in preparation for intestinal digestion, where enzymes break the particles even more. So it's important to note that pepsin is activated by hydrochloric acid, and we're going to test the ability of pepsin to break down a protein called albumin with or without hydrochloric acid being present. We're also going to test hydrochloric acid by itself today, and we'll be testing all of those at 37 degrees Celsius. So when you look at your procedure, it asks you to label 3 test tubes, 1, 2, and 3. So those have already been labeled here. So we have 1, 2, and 3. In test tube 1, we're going to put, in addition to albumin, we're going to put 3 mils of pepsin. In test tube 2, we'll put albumin and 3 mils of hydrochloric acid. And in test tube 3, we'll put albumin and 3 mils of gastric juice. And remember, the albumin is the protein that we're trying to break down. So I'll put that in first for each of the tubes. And then I'll put the solution that belongs in each tube. So we've got pepsin. I'm just going to fill it up to the little line that's drawn here on the tube, up to 3 mils to add it to the albumin. That's a little bit too much, that's okay. Pepsin. The next one is hydrochloric acid. And the last one is actual gastric juice. So a mixture of hydrochloric acid and pepsin together. Okay, now you need to make sure that they're mixed up together pretty well. Gotta get that. The albumin has a tendency to kind of clump together, so you just gotta make sure you mix it really well. If there's any on the sides, make sure you kind of smash it down in there and mix it up real good. It should look kind of hazy by the time you're done with it. For each of the 3 containers. Still got a couple chunks in there. Okay, so you can see they're all pretty hazy.